Hello, everybody. Today we are going to talk about something that you may have heard of called CRISPR-Cas9, sometimes just referred to as CRISPR. And over the past maybe 8 years, it's become clear that this is a very, very powerful tool for genome editing. Where did CRISPR come from and what does it do? That's what we'll be discussing in this video today. Okay, so to provide a perspective, it's important to think about when we think about genetics. Typically, the way genetic analysis works is it uses mutants, right? So to understand how genes work, we figure out what happens when they break. And there's some interesting perspective on how we obtained mutations. For many years in the 1920s, mutations were observed just by mutations were found just by finding them by chance. All right, so there's an interesting problem with this, and this little blurb of this interesting perspective on the sources of mutations says from the 1920s. This is in 1926 from a paper by Muller. After reviewing the repeated after reviewing the repeated failure of efforts by many workers to modify the mutation rate, Muller asked the question, do the preceding results mean? So they were having a hard time finding mutants. They were just only mutants they would get would be just ones they found by chance. Do the preceding results mean then that mutation is unique among biological processes in being itself outside of the reach of modification or control? That it occupies a position similar to that till recently characteristic of atomic transmutation in physical science in being purely spontaneous from within and not subject to influences commonly dealt with it? Must it be beyond our range of scientific tools? And it turns out that at the 20s it was frustrating to obtain mutations, but there was, during the time in which nuclear power was being developed and nuclear energy was being developed, people found out that radiation could be used as a form of mutation. And this is a really interesting paper that came out in the 1950s in which Ed Lewis used radiation to generate mutation. And there's some interesting quotes from this paper. He starts off by saying, a new method for detecting chromosomal rearrangements in Drosophila melanogaster has been applied to the problem of measuring the biological effects of ionizing radiations from nuclear detonations. All right, so the way that this work was done was basically nuclear bomb test sites were being used. They would strap basically a bunch of Drosophila in a vial to a post, and they would let the nuclear bomb go off for other reasons, that they weren't setting the nuclear bomb off for the mutational process. They were testing nuclear bombs, but they were using that as radiation. And there's an interesting quote in the acknowledgments. Finally, the author is indebted to Professor G.W. Beadle, who transported the flies to and from one of the nuclear detonation sites. So clearly, in the time from the 20s into the 50s, there was learned how mutations could be generated. And so that was with radiation, and of course also different types of chemicals. And what I'm showing here is a type of mustard that can cause chemical mutation, and other types of mutations. One's called EMS. So there's EMS right there, ethyl methylene sulfonate. And in 1968, it was used frequently to generate mutations. And so the point, though, however, is that in all of these efforts, whether through radiation or chemical mutagenesis, the mutations that were generated were random, right? So you take a Drosophila or you take a Neurospora bread mold or a mouse or whatever, and you expose them to EMS or irradiation, and the mutations that are generated just, there's no specific way of targeting mutations to certain genes. So the way that a genetic screen would be done is that, basically you'd have, in this particular case, a type of nematode here, usually probably C. elegans, and a mutation would be induced through what is called a mutagen, or typically a chemical, but could be a radiation. And then you collect the progeny that have been mutagenized, and then you cross them out one more time. And what you do is you screen for what are called rare mutant phenotypes. So this particular C. elegans could be behavior weird, or some weird developmental disorder, or unable to metabolize certain compounds, or something like that. And now you have a mutant in a particular pathway that's interesting to you from a biological perspective. But the mutations that are received in these progeny that are the progeny of the mutagenized parent, the mutations are randomly occurring. So a critical question then that was, basically bothering geneticists for many, 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 many decades is, how do we target specific mutations to the genome? And the way that we have figured out this most easily is something that we figured out in the past 10 years using something called CRISPR-Cas9. All right. So to actually explain how CRISPR works, it's important to recognize that immune systems have a specific function. You might be wondering, why are we talking about immune systems? And I'll tell you very shortly. So, what does an immune system fundamentally have to do? Well, the most important thing that an immune system does is, first, from the very beginning, be able to recognize cells that are from the same organism, shown here in blue. We call them self cells, right? So here's an immune cell. It needs to be able to distinguish the self cells from the non-self cells. So, for example, the cells of a parasite. And so we have this little barcode here that indicates, that there's, from this particular, the same organism that matches the barcode in the immune system. And so once self cells are recognized, there are efforts that will be made not to kill them. But when non-self cells are recognized, the immune system cell can basically target a directed attack at the invading non-self cell. And this could be any sort of parasite or infection. So the two parts, part one is recognizing self from non-self. And then part two, downstream of that, is once the non-self cells are identified, using various mechanisms to destroy the non-self cells. Okay, so step one, recognition. Step two, destroying the non-self cells. Now, that's a principle that you should understand when it comes to how an immune system would work. And before we actually talk about how that is related to mutagenesis, I'd like to give you some background on how we actually discovered CRISPR. And some of this would actually have been in the podcast that you listened to. So in the late 1980s, as genome sequencing or DNA sequencing began coming on board, obviously one of the first organisms whose genome was sequenced was E. coli. Right? And so this is a paper from the earlier period of the 1980s in which the E. coli genome was beginning to be sequenced. And one thing that was noticed is that there was an unusual arrangement, that's the title of this paper in fact, an unusual arrangement of repeated sequences in the E. coli K12 chromosome. And what we find here is that there are these repeated sequences, and this is a particular region of the genome. It tells you what the exact location is. But what the feature of it is that's interesting is that you have this repeated sequence. So G, G, A, G, T, C, so on and so forth. And you have it again and again and again. And in between, this is like sort of row by row, there are these other sequences that are not repeated. So there are tandem arrays, a repetitive sequence. And in between those repetitive sequences are not repeated sequences. Now, at the time, this observation was made, but no one knew what to make of it. And what these are called CRISPR arrays. And CRISPR stands for clustered, regularly interspaced or interspersed, short palindromic repeats. All right. And so what is found in these arrays, and this isn't just E. coli, this is other bacteria as well, is that there are these repeated sequences, and then there's this sort of like weird sort of spacer sequence that's not repetitive. Okay. So it turns out, and this is something you would have learned in the podcast, we can ask, okay, what are the sequences between the repetitive sequences? And once the ability to sequence many genomes was established in the late 90s and early 2000s, it quickly came to bear that people were able to actually analyze using computers the sequences inside these repetitive regions of the bacterial genome. And what it was found was that it contained phage. All right. So let me explain a little bit about what phage is. Actually, the full name for phage is bacteriophage. Phage means eat, and bacterio means bacteria. And so these are critters that you could all argue eat bacteria. So in this image up on top, what I have here is a bacterial cell. And what is being done is it's being attacked by phage. And these phage are basically viruses of bacteria. The viruses of the bacteria land on the surface of the bacteria. They inject the DNA sequences of themselves into the bacteria. The sequences then begin expressing proteins that are not bacterial proteins but are phage proteins. And at that point, they can hijack the bacteria and convert the bacteria from an organism that reproduces itself to an organism that turns into basically a factory for producing more phage. So in some ways, the phage hijack and take over the bacterial factory and just pump out more and more and more phage. And so actually, these phage may be landing on the bacteria or they could be exiting the bacteria. Now, this is not going to go well for the bacteria. If a bacteria has a phage infection, eventually the bacteria will explode and die. And so this is not good for the bacteria at all because it's basically taken over or parasitized by the phage. So this is how this occurs. Then the question becomes, well, how do these, let me just jump forward for a second, how do these phage DNA sequences end up inside these weird parts of the genome that are repetitive, end up inside the CRISPR array? All right, well, the CRISPR array actually has two parts. All right, there's the repetitive sequences in the phage, and on the other side, there are things called Cas genes, and Cas cleverly stands for CRISPR-associated sequence. All right, so it's not that clever. But there are genes nearby these CRISPR arrays that code for a lot of different interesting proteins. But how does the CRISPR array acquire the phage sequences of the bacteriophage? Well, the phage genome, as you can see here, is entering into the cell. And if it's recognized to be phage derived, it's recognized to be harmful. And it's broken up into bits, and those little chunks of the bacteriophage genome can be processed and inserted into the CRISPR array. Now, why the heck would a parasite such as bacteriophage be chopped up, the DNA of it be chopped up and inserted into the genome? It's kind of weird. It would basically be like, you're infected, for example, with HIV, and your body takes the DNA of HIV, chops it up, and inserts chunks of the HIV virus into your genome. Why would bacteria do something like this? Okay, so this goes back to what I just said before. I'll get to that question, answer that question in a moment. But nearby those repeated sequences are these Cas sequences called CRISPR-associated sequences. And bacteria have all sorts of different ones. And there's one particular very famous one called Cas9, what we'll talk about in a moment. Okay. Now, one thing that's interesting, I'll explain what CRISPR is doing and how it functions in a moment. But one thing that's really interesting is that not all bacteria have these systems, but many do. And so there are different types of Cas systems, all right, and shown in these different colors here. And if you look at E. coli should be in here somewhere. I can't find it right now. But you can see that there's all these different CRISPR systems in these different bacteria, such as Chlorobi and Thermatogi, and all the way down to, say, Chlamydia. There's a whole different assemblages of these different CRISPR systems that bacteria have inside their genomes. Okay, So why, going back to that question, is why does the genome take bits of um, DNA sequence in the virus and insert it into itself? Now, an important point that I need to make clear, which is is critical for understanding here, is that when you have a DNA or an RNA sequence, so either but it's still a nucleotide, right? So DNA or RNA. If you have a sequence, say for example in DNA, that goes like A, G, T, A, C, so A, G, T, A, C, the reverse of that, as we discussed the structure of DNA, would go, well, it goes G, A, T, T, G, C, so it would be G, A, so on and so forth. Well, I can't remember it off the top of my head, but you know what I mean. So you know that there's going to be the reverse complement sequence. Now, DNA sequences that are typically double-stranded wouldn't have this property, but a DNA sequence that is single-stranded has the ability to recognize DNA sequences that are also single-stranded, right? Because normally DNA is double-stranded. Actually, it's not just DNA that recognizes single-stranded DNA. RNA is also an excellent molecule that can hybridize to the sequences that correspond to it. Now, I want to be clear about this. DNA sequences that are of a particular sequence, like ATGC, right, aren't just going to recognize any random DNA sequence. They're going to recognize the DNA sequence that correspond to the reverse sequence of themselves. And that is very important because random DNA sequences won't recognize each other, and RNA sequences that are random won't recognize random DNA sequences. But if the RNA sequence corresponds to a DNA sequence that has the same sequence itself, you can get what is called an RNA DNA hybrid. Okay, that's an important thing to understand about nucleic acids. Well, it turns out that when the virus DNA, and there's actually another selfish element called plasmid DNA, when it gets recognized as parasitic, the DNA sequences get chopped up and put into these arrays. But what's interesting now is basically now you have these different colors, say for example, green or blue or purple or light blue. Those are sequences that correspond to the history of phage that has attacked that bacterial species. So, for example, in this particular case, this array indicates that over the history of this bacterial species, this species has been attacked by phage that have green sequences or blue sequences or purple sequences, so on and so forth. So there's almost a history of invasion embedded in the genome. Now, if those DNA sequences are transcribed, what that generates is RNA. Now, RNA molecules, and so have a great affinity to DNA sequences that they match to. Now, because this is an array, all right, and there's those repeated sequences, what you end up is a long transcript, and these little bubbles like that represent the sequences that are the repeat sequences in that array. And then the colored sequences represent the RNA version of the DNA sequence derived from phage. In addition to this array, which produces this long, what is called the cRNA, or pre-cRNA, there are also these different proteins called the Cas proteins or CRISPR-associated proteins. Okay. Now, the long RNA molecule that includes these little hairpins and also includes the sequences of the phage get chopped up again, and this is important. The chopping up of those small into smaller fragments basically is like a barcode recognizer for phage sequence. So what we have here is we have a previous phage which has green DNA, which corresponds to a specific sequence of DNA, but we also have purple RNA, which recognizes the sequences of a previous invasion. Now, what that does then is that sequence of RNA recognizes DNA from this purple phage, all right? and it's capable of, once it gets recognized, is chopping it up. So remember I said that there's two critical parts of an immune system. The first part is distinguishing self from non-self. So this is an identification of non-self. And then there's the destruction part, which is the cleavage. Okay, you can kind of get a little bit of a zoom in here. I'm not going to belabor this particular point too much, but you end up with this large CRISPR array, which makes what are called track RNAs. And the Cas enzyme, in this particular case known as Cas9, that is nearby the array, actually hybridizes or binds to the RNA sequence that recognizes phage. So what you end up with is a two-part machine that recognizes a sequence. It's an RNA that recognizes the DNA sequence, and it's a Cas9 protein that is going to then do something to the nucleic acid. Now, what Cas9 is what is called an RNA-guided nuclease. This is a crystal structure of Cas9. There are other Cases that do similar things, but this is the one that's most commonly used. It's an RNA-guided nuclease. So a nuclease means chops up nucleic acids. And RNA-guided means that if an RNA sits in it, it will guide that Cas9 to specifically DNA sequences, in this case, that are corresponding to the RNA. Okay, so what you end up now is with a two-part system, all right? You have, and this is another zoom in, you have the Cas9 protein here, and then you have a RNA sequence, and Cas9 is able to open up the DNA and then probe the DNA sequence with this RNA molecule. In this particular case, what we have is a match. So this might be A, T, G, C, and then the other side would be G, C, T, A, T. All right, so the sequence would match. And there are some requirements. There's this thing called the PAM sequence that is a requirement for Cas9 to recognize it. But there's this thing that we then generate called a guide RNA, and we can synthesize these guide RNAs in our lab. So they don't have to be, and this is important, they don't have to be recognizing phage. Now, if you were to program it with a phage RNA, it would recognize phage. But what if instead of basically using the phage guide RNAs, you generate your own guide RNA? And as a matter of fact, what if you generate a guide RNA that targets a particular genome, a particular gene that you're interested in? Okay. The other thing that this Cas9 does, in addition to holding this RNA and using it to recognize sequences, is it has that nuclease activity. And what it does is it shows these little scissors. It will cut the underlying DNA sequence at that position. Now, you might be wondering to yourself, wait, that doesn't seem very precise. And you know what? You would be right. It's not an extremely precise method. It's precise in recognizing sequences, but the way in which it cuts is not always precise. But in some ways that might be good. Okay, this is just another zoom in of how the sequence matching would work. There's going to be a sequence recognition, and then there's going to be this PAM sequence, and this guide RNA that is in complex with Cas9 would cut at this position. So what's the significance of cutting DNA? Well, the important thing is that the cutting of the DNA sequence is a specific DNA sequence in the genome. So what are the outcomes? Now, if you just express one guide RNA, again, not targeting towards phage, but targeting to any gene in the genome, a cut will occur. And so then the DNA has to repair itself. And there are several different ways that DNA can repair itself after it has been cut. Sometimes these breaks just happen by chance alone, actually. So one way that DNA sequences can get repaired is that the two ends that are broken just get shoved back together, and this is called non-homologous end joining. The ends are joined, and they're called non-homologous because the sequences of one end don't match the sequences of the other end. Now, the significance of this is that if this non-homologous end joining happens inside a gene, it can break the gene, right? Because genes have a coding sequence, right? You have a start codon, and you have all the three nucleotide codons for all of the amino acids. If a break occurs, that is what is called a frameshift mutation, and say, for example, one base pair or two base pairs or six, well, no, not six, right? One base pair or two base pair, or say five or seven, any insertion or deletion of that number of base pairs will upset the reading frame. Now, if you have an insertion of 3 or 6 or 9 or deletion of that size, that will not upset the reading frame. So if you upset the reading frame, you break a gene. Okay, that again might not seem very important, but what you can do and understand is that going back to the beginning of this lecture, the folks who were doing genetics had a heck of a time figuring out how you could generate mutants. And what this allows you to do is generate a mutant to a specific sequence. Now, how do you know which genes to target? Well, the important part here is that we have the genomes of these organisms that we study. We have the genomes already sequenced. So we obtain the sequence of the gene of interest, then we knock that gene out, and then we have a new mutation. Now, I said non-homologous end joining. Here up in this little table, you have non-homologous end joining, which can lead to gene disruption or via small insertions or deletions. Another thing that can happen is that inversions can happen because the DNA tries to repair itself in some sort of weird way. You can have very large deletions. So there's all sorts of ways in which non-homologous end joining can actually lead to disruption. But another thing that happens in the cell is that when there is a stretch of DNA sequence that is broken, oftentimes it repairs itself off of sequences that look the same. Now, all of our chromosomes come in pairs, except for the sex chromosomes. So if you have a mutation on chromosome 2, the one you got from your mom, and there's a break, it can repair itself from the other copy in the cell. And that particular case would be the chromosome corresponding to the one that you got from your father. And so if there's a break, and so that's what I'm showing here, if there's a break here or a break here, the sequence from the non-broken chromosome, here shown with an orange arrow, or here shown with a purple arrow, the sequence can be used to fill in the gap. And what this ends up doing is actually adding sequence to the broken piece of chromosome. Now, what is the significance of homology-directed repair? Why don't we just use non-homologous end joining? The reason homology-directed repair is important is you can provide a sequence that corresponds to the sequences that you know you want to target. And so in this particular case, you have a target strand. Here's the Cas9 complex. In green is the guide RNA. It recognizes a very specific part of the genome, of one gene perhaps, and it breaks it. Now, if it's broken in this way, this is called a double-stranded break, a researcher can provide sequences that correspond to the left and sequences that correspond to the right. They can synthesize this DNA molecule in the lab. Then, in the middle, they have a desired insert. Now, I won't go through the details of how the homologous repair works, but basically what it does is the blue sequences recognize each other, the blue sequences recognize each other, and the red sequences just dropped right in to the middle of a particular gene. Now, before I said CRISPR-Cas can make mutations, but here, in some ways, if you think about it, it can also repair mutations. Because if this sequence here is defective, you can drop in a repaired sequence for a gene. You can see now how that might be very powerful, both in the lab, but also for cases of human disease that are caused by defective genes. Okay, so different examples have been provided in mice for doing this type of work. One can actually generate mutations that cause defects in vision, that have cataracts in mice, and one can do two alternative methods. One can actually inject, in this particular case, the Cas9 enzyme and the guide RNA and target the gene that's broken. So there's this mutation here that causes cataracts. Now, if you don't provide any homologous template, what you do is you get HDR, that means homology directed repair, based on the endogenous wild type allele. So in this particular case, there was a deletion that was defective and gave these mice cataracts. Well, if you cut that, the corresponding wild type allele on the other chromosome can be used as a repair, and this can cause offspring to be repaired. Now, sometimes you might ask yourself, like, well, will I always get a good repair product? Well, after making a break, will the sequence corresponding to another gene in the genome come and fill that in? And instead of actually letting that happen, you can also generate what is called an oligonucleotide. You can synthesize the DNA sequence. That's sort of what I implied here. This would be a synthesized DNA sequence. And that synthesized DNA sequence would recognize the break after Cas9 cuts there, and that sequence would flood the system of breaks and actually lead to an insertion of the red sequence in this particular case. Okay, now, so two things that we said that CRISPR can do. One, it can target mutations and break genes. That might be important. The other thing is it can actually recognize sequences in the genome and provide, through homologous repair, repaired sequences. We'll talk about the technical significance of that substantially next week in class. But CRISPR can do many, many other things, right? Because it directs an enzyme that cuts to a specific region in the genome. It can direct enzymes to not just cut, but it can direct enzymes to do all sorts of things. So, CRISPR can be used to target certain genes to identify proteins that bind a sequence. It can target certain genes to become activated because instead of basically breaking coding sequences, it can break regulatory sequences. It can target fluorescent proteins to genes to allow you to see where the genes are in the cell or in the nucleus. And it can also generate chromosomal rearrangements. Okay. So a couple points I'm going to end with is, as we know, this is potentially a controversial method. There's a distinction that must be made is if you are going to edit a given person's what I call soma, or others call soma, or the tissues of an individual, versus editing what you would call the germline. Okay, and people have generated germline mutations in human embryos. As a matter of fact, last year, you might remember, in China, a researcher very controversially edited the genomes of at least two different babies. Now, it may be possible that someday in the future, it might be considered important to edit your children's genome. On the right hand here, I have a bunch of mutations that basically people are known to survive. And if you have mutations in these are not generated by humans. These are just randomly segregating in the human population. If you have these mutations, for example, if you have a mutation in CCR5, if you have a mutation in PCSK9, this individual has out in the world, they don't have that functional gene. They have very low rates of coronary disease. And individuals that are mutant for CCR5, those individuals cannot get HIV. So if you knock out these different genes, and we know this because we know that there are people out there just randomly who have these mutations, they have potentially a lot of health benefits by getting rid of these genes. You might be saying to yourself, wait a minute here, why would you knock a gene out? Maybe that gene is important for something. And that's a good point. That's a very good point that people sometimes forget. One thing that you can do with CRISPR, and we'll talk about this extensively in the next class, is you can actually modify, say, for example, pig genomes and turn them into sort of like genomes that are not going to be problematic for human immune responses. Okay, so a huge problem is obtaining donated organs for people who have organ failure. Now, one way that that's done is humans can donate their organs when they die to another human, for example, heart transplant or lung transplant or liver transplant. A problem with that, though, is that one, it's hard to get the material because there's not enough people who donate their tissues. That's part one. But number two, a lot of those, if you don't have a good immune match, if you get a heart transplant from somebody and you're not careful, it could induce a harmful immune response. And because of that, individuals that have received organs from other humans are on drugs for the rest of their lives that suppress their immune system so that the immune system doesn't attack the donated organ. Here we get back to the self versus non-self thing. Well, what if you could take the pig genome and knock out all the genes that you're concerned about that would induce an immune response in humans? And people have actually done that. They're endogenous retroviruses. They have a somewhat unfortunate name for them. They're called porcine, pork, pig, porcine endogenous retroviruses, and their unfortunate name is they're called PERVs. But anyway, people have generated pigs that have knocked out all of the viruses that exist within the pig genome. And it's possible that we may be able to use pigs now by doing continued modification with CRISPR to knock out genes that elicit an immune response in humans to generate, therefore, organs that will be functional for humans to receive. A last thing I want to mention is that another thing that people have developed applications for with CRISPR is to modify the genomes not one by one of an organism or a species, but in a sort of chain reaction way in which you could actually drive a gene modification through an organism. Now, what if at a particular region of the genome, you in fact encode all the machinery itself into the genome itself? So here's an allele, right? And this allele you insert, and you could do this with mosquitoes or some sort of viral vector that is going to cause problems for, say, for example, malaria transmission. What if you actually drop into a gene, and this gene might be something that's critical for spreading viruses in, say, ticks or whatever? What if you drop inside that gene the Cas9 enzyme, a sequence that encodes for a guide RNA, and a cargo sequence that you might do something for else? Well, the guide RNA in this particular case recognizes the other allele, the other chromosome. And so what it will do is, you have, you're heterozygous, you have one copy from mom and one copy from dad. Well, the one copy from mom actually cuts the copy from dad, and because of homologous repair, it copies this sequence into this individual. So now you've kind of messed with Mendel at this point, because now something that is heterozygous has been converted into something that's homozygous. If you let this process run out, what you can end up doing is, under normal circumstances, a low frequency allele will stay low frequency in a population. But when you have this form of what is called gene drive, in which a heterozygous allele can actually convert the other allele into its type, that gene drive can spread that copy throughout the entire species. And you could drive a variation in the species, and perhaps that variation might be, for example, the inability to transmit malaria. So you can see how then you can shape an entire species, and that might be beneficial. So that's the end of the show, and we will talk about the technical aspects of CRISPR and make sure everybody understands in class on Tuesday.